So it's a real pleasure to be here, and I really feel honored to be part of the symposium, honoring Jim and his outstanding contributions to environmental chemistry in the atmosphere. So congratulations, Jim. Very well deserved. Very well deserved. So atmospheric chemistry has a lot of different approaches. So we need them all. So a laboratory experiment, and I've lumped theory in with this, field measurements to see if we actually see in air what we think we should based on the laboratory experiments and the theory, and then modeling from local to global scales that really pulls it all together. So I was fortunate to be co-chair of a National Academy panel a few years ago on the future of atmospheric chemistry research, and co-chaired with Bob Duth. And one of the things that came out of that was the real emphasis on developing a predictive capability. So pulling all this together in such a way that we can, with confidence, say what we should do to address air quality and climate change. So today, I'm going to focus on the importance of understanding interactions at a molecular scale for developing that kind of predictive capability. So if we go back to the early days of photochemical air pollution, Ari Hagenschmidt at Caltech. So he published, I think it was in a Caltech magazine in 1950, and then subsequently in a number of papers, the basic chemistry involved with photochemical air pollution. So that it was volatile organics and oxides of nitrogen that were really the key ingredients to the whole thing. And then in the 1960s, the photochemists got involved. And so people like Jim and Jack started applying their physical chemistry, photochemistry to these problems. And they, for example, spent a lot of time on singlet oxygen as a possible oxidant for VOC, because at that point we didn't know what the oxidant was. Well, very interesting. 1970, Greiner, who was at Los Alamos, published some papers on OH kinetics with a variety of organics like alkanes. So this is the Journal of Chemical Physics. And he had no idea, I think, that it might be important in the atmosphere, but this really opened up the whole field. And from there, people took it and showed that, in fact, OH was one of the key drivers. About that time, people were also getting interested in the stratosphere. And our very own Jim Anderson here was working with Fred Kaufman and looking at stratospheric chemistry. And of course, that led to the CFC and NOx identification of issues in the stratosphere. So I'm going to give you a few examples from this lab of a molecular level approach to issues in atmospheric chemistry and air quality. So we've worked a lot on sea salt particles in the past, and we were very fortunate to work with theorists Benny Gerber and Pavel Jungwirth and Doug Tobias. And they showed using molecular dynamic simulations that chloride ions were readily available at the interface, and you could get a lot of really interesting and different interface chemistry that turned out to be the key to explaining our experimental results. Another area is particle formation from methane sulfonic acid. So this had been ruled out as contributing to particle formation in air, but that was before we really recognized the importance of amines. And so we've done a lot of work on looking at amine effects on particle formation, and again, working with theorists and showing that you can get clusters like this that are stable enough that they then form the seed to grow on and form particles. But today I'm going to focus on two areas, other areas. So one is unusual neonicotinoid photochemistry, and the other one is growth mechanisms of highly viscous secondary organic aerosol. And it turns out that gets into the area of what's the surface composition of these things versus the bulk. So let me start with the pesticides. And as I go along, as I said in my cover slide, it's me and a ton of other people. It's really mainly the ton of other people's work I'm going to talk about. So these are some of the folks who have been involved with the neonicotinoid photochemistry, and the reason for the B's on the cover here will become obvious in just a moment. So the neonicotinoids got their names from the fact they bind to the nicotine receptor. And there are three flavors of neonicotinoids. So there's, whoops, got a little carried away there. The nitroguanidines are C double bond N, NO2. Imidacloprid is the one that's used most widely around the world. It was introduced in 1991. Then there are cyano compounds, and then there are these nitroenamines. So an amine double bond NO2 nitin-pyram is typical of that. So if you put these drops on the back of your dog for flea control, that probably has nitin-pyram in it. So the reason that, that these have become so prominent is that they have very low mammalian toxicity in contrast to a lot of the other pesticides that were used. So they've been used very heavily for seed treatment. So they put them as a coating on seeds, and they're systemic. So as the seeds grow into a plant, these things are in the plant, and then when aphids and other biting and sucking insects hit the leaves, they get the pesticide. But as is true of many, many environmental issues, you know, not everything, in fact, only a small portion of it ends up being effective, and less than 20% actually ends up being absorbed, and the rest ends up in the environment in various ways. So they're low mammalian toxicity, but over the years, it's pretty clear they're associated with impacts on pollinators and the bee colony collapse, for example, which, you know, we rely on pollinators as for our food sources largely. So this is a nice example, I thought, of how widely these things get distributed. This was a citizen science project, and they had people send in honey samples from all around the world. So the blue is the imidacloprid, and you'll see that it's a major contributor everywhere but Europe. Europe had actually regulated these things starting in 2014. It regulated some of them, and that was imidacloprid, so that's why you don't see that showing up here. But 3/4 of all the samples that were sent in from around the world had at least one neonicotinoid, and almost half had more than one neonicotinoid. So they're really widely spread. So what about their photochemistry? So these are absorption spectra of 3 different types. And the actinic region, of course, is 290 on up. So if you look at imidacloprid, you'll see it's got a little tail here in the actinic region, so you'd expect it to do some photochemistry. The cyano compounds don't, so they wouldn't do tropospheric photochemistry. The nitin-pyram actually has a pretty strong absorption out here, but I will mention that that's really dependent on whether it's a solid or whether it's in water and what the pH of water is. So not surprisingly, these two groups have NO2 groups you'd expect to be the chromophore. So we've looked at imidacloprid. In fact, we've looked at a whole series of them, but I'll just show you some data on imidacloprid. And asked what the products are. So these are mass spec, DART mass spec data of imidacloprid before photolysis, and you see the M+H here at 256, 258. And then the photolyzed sample has a bunch of peaks around 212. The 195 is the caffeine we added for calibration, so that doesn't count. So if you look at the possible products, what you see is that the peaks around 211 really correspond to 2 sets of products. One is the urea product. So you've chopped off this and you've made it a double bond O. And then the other product is the des-nitro, it's C double bond N. And it turns out this has been shown by other people to be more toxic than the parent compound. So it's really a good example. The same thing story as malathion, where the parent compound may have low toxicity, but you really need to worry about what happens when they're environment and what they actually form and the toxicity of those compounds. So more interesting from a fundamental chemistry point of view was the gas phase. So we looked at the gas phase expecting to see NO2. And this is the infrared spectrum as a function of time, what we saw here. It turns out it's not NO2, it's N2O. And I have to say, we really scratched our heads a lot about this. But, you know, this is a really characteristic signature. You're not going to misidentify this. And it turns out all the nitroguanidines have a high yield of N2O. It's basically with an experimental error of unity. So we talked to our theoretical colleagues, Benny Gerber, and he has a wonderful postdoctor at Chemish, and they did an isolated molecule calculation. He came back and he said, well, it's going to form NO2. I said, yeah, that's what I thought, but it's N2O. And so we got thinking about it and realized that in a solid, when you break the bond, when you photolyze this and break off the NO2, these things are held together in a solid. You know, it's not like the gas phase where they can fly apart. And so because they're held together in a solid, they recombine. And and so when they recombine, you're getting that bond energy back again. So what Benny and Dorit did is said, let's take the molecule, put that energy into the ground electronic state, but the vibrational modes, distribute it statistically amongst the vibrational modes, and ask what molecular gymnastics this would sort of cause. And so what they predicted is this excited molecule would have the oxygen of the NO2 start interacting with the hydrogen of the amine, basically break off an incipient OH, which then attacks the carbon, and you end up with this molecule and N2O, and then this rearranges to give you the urea. So, so theory was really extraordinarily useful in understanding this, and this was something that we could never have predicted a priori. So they went through all the steps, all the energetics. I'm not going to go through this. See this paper if you're interested. But the lifetime, you can calculate, say at a solar zenith angle of 35 degrees is about 16 hours. So these things don't last that long in the atmosphere, and you're going to form these products, some of which are more toxic, and then N2O. So, so what we've learned from this part of the neonicotinoids, we've done a lot of other different studies, is that they do absorb in the actinic region, and they have somewhat short lifetimes. You get products that are known to be more toxic than the parent compound. You get these really unusual photochemistry that generates greenhouse gas, which we couldn't have guessed. Now, the amount of N2O you get out of these things compared to what comes from processes in soil and other things is not important. So this is more of a basic chemistry interest. And then the other thing that we realized is that these are really complex molecules. They've got amines, they've got aromatic groups, NO2, some of them double bonds. So we now have a project going on to try to use model compounds and pick out an individual group one at a time and then put them together in the molecule and see how that affects their reactivity. So if you're interested in that, Xinqi Wang is giving a talk tomorrow, 3:05. So let me hit the second part of this, and that's secondary organic aerosol. So many of you people in this room, I'm sure, have done this experiment. I think, in fact, Stan, I think you and Mike did this video of adding high concentrations of ozone to alpha pine, and you get this white smoke, right? These particles. So it had been assumed for many years that these were little liquid particles, like oily liquid particles. But about 10 years ago, a Finnish group observed something in impactors that we have also observed, and that is when you take an impactor, and Carla Kidd did this work, so you put particles in, and they're forced through these small holes. If they're big enough and under the right flow conditions, the larger particles impact, and in this case, we have an infrared crystal, attenuated total reflectance crystal, so we can pull it out and look at the infrared. But they impact the smaller particles. Get carried away in the gas stream. So if all's right with the world, if they impact, you should get a stream, set of particles collected underneath those holes. But if they're solid, what happens is they can bounce. So they hit the surface and they don't stick. And they can either bounce back out into the stream so you don't see them, or if your crystal's wide enough, they'll bounce and be captured on the crystal. So this is our large volume, slow flow reactor that we use for these studies. And we know that SOA is not a liquid under these conditions. And these are, I'll say right now, this is no water right now. It's dry system. So they're high viscosity semi-solids, and we can see that from the impactor patterns. So this is SOA, and the holes are right here. And then all this stuff we call a cloud around it is where the SOA has bounced. And then if you put grease on the impactor, which people do a lot in ambient air measurements, it just sort of deep sixes into this grease and doesn't bounce. For comparison, this is what a liquid looks like if you put it through the impactor and it just hits and sticks. In fact, you can even see here where the machinist was off center in one of the holes here. So we always think what we're doing is really new. Nobody's seen this before. Really interesting. You go back to 1975, and these were studies done a few years earlier in LA by Hussar and Shu, and so they show these electron microscopy images of these particles, and they describe them as looking like rubber balls, flattened rubber balls, and so plus ça change, plus c'est la même chose, same thing, right? Or in the words of Yogi Berra, it's déjà vu all over again. So we really need to worry about phase and viscosity and diffusion. So this is a kind of what's become a classic figure from Koop et al. So it shows viscosity along here and then corresponding diffusion coefficients, assuming the Stokes-Einstein equation, which applies for a lot of things, not everything, not under all conditions, but it's kind of a general marker. And they've got up here the different things: water, olive oil, going all the way to glass marble. So in this region of viscosities, they're liquids, and then you move into solids and semi-solids and solids. So if you have a 100 nanometer particle that's a liquid, diffusion times are of the order of milliseconds, 1 to 10 milliseconds. But if you're up in this region, it can be, hours, days, months, years. So exchange of gas with these particles really depends strongly on what the phase of these things is and how fast things diffuse in and out. So, so what happens in air? So this is from Manabu Shirawa's modeling work. So he asked, what would the phase be of particles around the world under different conditions? And so this is the top one is at the surface, and then this is at about 5,000 feet, and then 18,000 feet. And the red corresponds to solid, the yellow semi-solid. And you can see under many conditions in many places, the particles are really high viscosity under large sections of the atmosphere. So, so what does that mean for growth mechanisms? So we've assumed in the past these are liquid. And so low viscosity and the VOC oxidation products are going to partition into the particles, and you're going to have a kind of quasi-equilibrium distribution characterized by a partition coefficient. But if they're not low viscosity, if they're high viscosity semi-solids, the oxidation products come in and they're going to absorb on the surface. And they're not going to diffuse in very quickly. And you're going to keep having more and more of these oxidation products come in. So this is what's called condensational growth. And in that case, what we need are uptake coefficients. So what we're concerned with is how do the particles grow, which mechanism, and does it matter for model predictions? And then are the particles homogeneous like you'd expect in this case, or are they heterogeneous in 3 dimensions and kind of built like an onion because of the growth process? So a group a few years ago did some modeling to see if they could explain their measurements in Huhtala, Finland. So these are the banana plots. So it's the diameter is a function of time. So you see this red here, this shaped like a banana. It's particles that are forming and growing with time. And what they did is asked, does the quasi-equilibrium model work? And this is what they predicted, and it doesn't do a very good job of explaining the experimental results. If they put 100% of the condensational growth, it gave a pretty good representation, and even 50 did. So somewhere between 50 and 100% of this kinetically controlled uptake needed to be included to explain the results. So it seems as though it's something we should be considering. So the question is, what determines it? So there's been a big emphasis on vapor pressure of the organics as being a determinant of what contributes to particle formation and growth. The question is that the only determinant? And of course, vapor pressure is correlated to molecular structure. But what's the molecular structure of the gas on the particle surface, and does it matter? So our approach is to measure the uptake of molecular markers during particle growth compared to equilibrium. And then we get into this issue of the surface and what its molecular composition is. So Alison van der Waal did this work as part of her thesis, actually her whole thesis. She did an amazing job, and with help from Veronique and Lisa here. So we used 3 molecular markers. And the first one is commercially available, so it's a long alkyl chain nitrate. The other 2 had to be synthesized. So the second one is a propyl nitrate, so it's got a much shorter alkyl chain, but it's got an OH group. And then the third one has the long chain similar to the first molecule, but it also has an OH group. So these 2 molecules, if you calculate their vapor pressures, they're about the same, but they're clearly structurally quite different. These 2 molecules, if you compare EHN and what we're calling HHN, they have the same hydrocarbon chains, but the vapor pressure of this one is 8 times 10 to the minus 6 atmospheres calculated, so much lower vapor pressure, not surprisingly, with the OH group on it. And then if you compare these 2, they have similar hydrogen bonding capabilities. So with these 3, we're trying to cover the territory in terms of what the molecular forces might be. So we've taken a number of approaches, and I'll just give you a few examples of the data. So in one approach, we take ozone with alpha-pinene, and we discovered early on that because it generates OH, it also reacts with the markers. So a lot of our experiments were done adding cyclohexane as an OH trap. And we take the SOA and we can impact it on the ATR crystal, and then we can pass organic nitrates over it and watch the uptake in the thin film of secondary organic aerosol and get partitioning coefficients. So this is an equilibrium partitioning type experiment. Then the other set I'm going to talk about, so that's called what we call Series A. Series CD here, we form it SOA in our large flow tube and take out the OH, and then we look at the particle composition by either aerosol mass spectrometry, or we can impact it and look at it by FTIR. So what we're after here is what's the uptake if the organic nitrate, the tracer, is there while the particles are being formed. So in the first case, you know, the action's all over for SOA formation and you're just exposing it afterwards. In this case, you are actually having it there while the particles are being formed. So if you look at the partition coefficient, you see some really interesting things. So the left side is Series A, and these are partition coefficients for EHN, the propyl nitrate and the hexyl nitrate. And you can see one of the first things is, although the vapor pressures of these two molecules are similar, really different partitioning. So it really emphasizes the importance of molecular structure in that OH group. But the other thing is, things on the left are all equilibrium partitioning. The ones on the right were the partitioning, quote unquote, when we used our flow tube to actually have the organic nitrate there during particle growth. So you're having particles formed from these low volatility compounds, and they're taking up this organic nitrate at the same time. And it's very clear that, that the amount of nitrate that gets taken up is higher when it's there during particle growth rather than afterwards. So the other question is, what about uptake coefficients? So we were able to extract from the data uptake coefficient estimates. And in this series of experiments, we kept the ozone concentration constant, increased the alpha pinene. And what that does is it, of course, pushes the kinetics and the chemistry faster. So you have, you're forming the compounds that will form particles a lot faster. You're getting higher concentrations of those. But we kept the organic nitrate constant. So we're just forcing the SOA part of it to go faster. And what you see is that the uptake coefficients actually increase with this alpha pinene concentration. And interesting, again, 2HN and HPN, similar vapor pressures, have very different uptake coefficients. And again, having the OH on here really makes a difference. So the fact that this uptake isn't constant and it seems to increase with the increased high molecular weight contributions suggests to us that there's a burying mechanism involved in this. And I'll talk about that in just a second. So, and then this has a much larger uptake despite similar vapor pressures. So this is our view of the burying mechanism right now. So you take the gas in and it hits the surface, and it can scatter, or it can be absorbed and be on the surface for a certain period of time and then come back out. So it's kind of the Gill-Nathanson sort of vision of interface chemistry. And then this if it's on there long enough, can get taken up into the bulk. But what we are proposing to explain our results is that the burying species, so these are the high molecular weight things, the things that the alpha pinene ozone reaction is forming that are coming to the surface and they're going to add to the particle and grow it, that if while this is on the surface, this comes in, it can actually bury it. And that helps to have it taken up into the particle bulk. But if that's the case, if we want to really probe this, we need to know how long these things stick on the surface. And this really sends you down a rabbit hole, I'll tell you. So there's a whole bunch of different things you need to know then. But what we've learned so far is that the molecular structure does matter, even if they have similar vapor pressures. The uptake of gases may occur in part by a burying mechanism. And then this seems to result in much higher concentrations in the particles than you would expect based on equilibrium. But what we don't know is a lot. So the residence time of the gases on the surface is going to matter, because if they have really like microsecond residence times and they come off, they're not going to have a chance to get buried. So we need to know the rate of adsorption to the surface. We need to know the binding energy. the molecular nature of the surface. And our overall goal here really is to ask, can we develop a predictive capability where we say you have a gas that's polar, you have a surface that's polar, that will result in these attractive forces and this growth mechanism and the amount of growth. Or if you have nonpolar and nonpolar, much weaker attractive forces, and it would grow it much less. So we're hoping that we can get that kind of general correlation by looking at this. So to get the residence time in the surface, we're in the process of building a TPD apparatus. In fact, all the parts arrived on Friday from the machine shop finally. And Yixin Li here is working on it. I get in the lab occasionally, and it scares the hell out of my group, I think, right? They'll say, yes, yes. So here's Mike and I underneath the apparatus trying to get the turbo pumps fixed. So these are the kind of data that we're expecting to get. This is from a paper by Bruce Kay. So he put benzene on graphene, and the first layer, if there's just one layer, you measure the desorption rate as you heat up the system, and about 160 the benzene is coming off. But if you put multi-layers of benzene on, then you really have benzene adsorbed to benzene, and the attractive forces are smaller, and then you get a peak coming off that reflects that multi-layer coverage. So you can analyze this and get desorption energies and prefactors and so on. So that's what we're planning on doing for a series of well-defined surfaces and molecules. So the last part of this is we need to know the surface composition, because if we're going to get these attractive forces, we know the gas, we know what gas we're putting in, but what's that surface look like? And there are lots and lots of nifty techniques to measure particle composition in air now, right? Bulk compositions, there's aerosol mass spectrometry, there's laser ablation, tons of good ways of doing it. But we really need to get at that surface. So we really want to peel this like an onion. And now, people would look at me and say, are you crazy? Do you really think these will have a different surface? And so I'm going to give you an example that when I show it to atmospheric audiences, they go, oh, this has nothing to do with atmospheric. It doesn't directly to an atmospheric system, but I think the principle is there that I think is worth thinking about. So we took self-assembled monolayers and reacted it with ozone. And terminal double bond, so ozone, I thought, would chop off the double bond and you get acids and aldehydes. And Teresa McIntyre, who was doing this work, did AFM to make sure the films were good. And then she came back and she said, this is what, she did AFM after the reaction. And I thought, at our resolution, we're not going to see any difference. But she came back, and this is what she saw, and I thought, did you get some dirt on this on the way to the AFM? But she's very careful. That wasn't the case. And in fact, if you do single particle FTIR on it, you see all kinds of peaks, down in this, 1600, 1700 region and up above 3000 that clearly reflect highly oxidized functional groups in these. So these are organic. So we also did nanosims and sort of drilled in from the top down to get O and C. And so the surface is over here on the left. Forget this first point, that's water on the surface. But as you drill in, what you see is that the O to C ratio actually goes up as you drill from the surface inside the particle. And so what it suggests is a model like this, where all the polar groups are buried inside a hydrophobic shell. Now, I said this is not an atmospheric particle, but I think the principle is something that we need to think about in terms of the composition of these highly viscous particles. So we've done a number of approaches to trying to probe the surface versus the bulk. And we've done extractive electrospray and shown under some conditions that probes the surface preferentially. Another way of going about it is sonic spray ionization. And so you put a solvent through the center, and then outside you have a gas, nitrogen at supersonic speeds. And Lisa Wingen has done this work. And so the friction from the sheath gas moving over the solvent generates charged solvent droplets. So it's kind of like almost an ESI experiment. So it's soft ionization. And so if we bring in a beam of particles, if you bring them up here very close to where the solvent is coming out of the nebulizer tip, or the tip here, it turns out they just dissolve, and what you see is what looks like the bulk composition of the particles. But if you put it way downstream where the solvent's evaporated and you're just left with charged species and clusters, it turns out you actually just see the surface. And so this is just one set of data. So this was a glutaric acid core, and we put a malonic acid coating on it to get a core shell structure. And this was in the orthogonal mode we called, like way downstream. So what you see is you increase the coating thickness of the malonic acid. First, when you don't have a coating or it's very thin, you don't see the malonic acid at all, but you see the glutaric acid core. And then as you increase the coating thickness, you see more and more of that coating. But it's really shielding the core, and so the core drops off. And it turns out we think we can probe about 3.5 nanometers of the surface using this technique. So the last technique, and this is one that we're working on now, is what's called vacuum or magic ionization. So Yiming, Jin, and Lisa are working on this. So we take glutaric acid again and we atomize it. And we've got a mass spectrometer where we take out the ion source. And so we've got a waters and we just take off the ion source. You have the front end here. So these species are coming in, the particles are coming into the inlet. They're going into this heated block, and we can change the temperature from 30 to 150. So when you do that, this is just glutaric acid, you know, no voltage, no heat, no light, no ion source except for cosmic rays in the room. So the bottom is the total ion chromatogram, and the left is at 30 degrees, and then we've increased the temperature. So you're getting signal from these particles, and in between we turn the particle beam off, and there's nothing there. And the great thing about these kinds of techniques is, you know, the background is zero because you don't have any kind of background that you're ionizing. And so you can live with small signals. Above that are the mass spec that were taken. So glutaric acid has a molecular weight of 132. So 133 is the protonated adduct. And then all these other peaks we can identify as being sodium, ammonium, and so on. So it's clearly glutaric acid. Now, what happens if you take secondary organic aerosol and you put it in? Turns out nothing, basically. You get very little signal at all. But if you take glutaric acid and then you coat it with secondary organic aerosol, so you put alpha pinene and ozone in with the glutaric acid particles and put them in the mass spectrometer, you get a beautiful spectrum here. And there's the glutaric acid, but there's also, you can identify the usual pinic acid, pinonic acid, and all these high molecular weight products. Again, no heat, no light, no voltage, and you're getting this SOA spectrum by putting it on top of glutaric acid. So Yiming is a cat person, and as she says, it's not, I'm not saying it's magic, but it really is magic. So the temperature dependence we've looked at, and the atomized particles, we know we get out of atomizers, the particles are charged, have some charge. So then we tried self-nucleation. So you just heat up the glutaric acid and let it self-nucleate. And in that case, you get much lower signals. And then there's an ion gun you can buy off Amazon that's used for taking static off the old vinyl records. And if you pull it in, you get one charge. You let it go, you get the other charge. So you can charge things selectively. So if you take the self-nucleated and hit them with an ion gun, again, you bring the signals back up. So, so we've looked at the temperature dependence of this. And the self-nucleation down here has a temperature dependence that indicates a sort of effective activation energy of 92 kilojoules. The atomized is 21, and then the winner is the self-nucleation with the ion gun. So it's pretty clear that, you know, the more charges you have on there, the less energy you need to have this happen. But what's really interesting is that the sublimation energy for glutaric acid is 120 kilojoules per mole. And these are all less than that. And yet we know that sublimation is playing a really key role in this whole process. So Sarah Trimpen at Wayne State has really been the person who started this many years ago. She said she had a very difficult time getting through review, saying she was seeing things without any kind of energy source. And I think right now, personally, I don't think I really understand what's happening. And so that's one thing we're working on now. So what we've learned is that mass spectrometry techniques do have promise for peeling these things like an onion. And that's part of the key that we really need to understand particle growth for these high viscosity particles. So I hope I have convinced you that laboratory experiments and theory are a very central part of atmospheric chemistry. And that understanding these is really central to developing a predictive capability. So with that, let me acknowledge my research group. This was our department dinner pre-COVID when we could get together. and then a wonderful group of colleagues at AirUCI that I'm really privileged to work with. National Science Foundation has been a faithful supporter of our work over many years, which we really appreciate. The Army Research Office has provided some support for this and also for equipment. And then I want to thank the Dreyfus Foundation for their really support of environmental chemistry over many years before it was really fashionable. And it really has made a big impact, I think. And I was telling Scott that I was the beneficiary of Dreyfus money many years ago to develop a new analytical lab at the junior-senior level that was based around analysis of environmental samples. And those were all published in JChem Ed with grad students as co-authors. And so, you know, as Scott said last night, you know, Dreyfus Foundation punches above its weight, really. And so with that, let me thank you very much for your attention.